All right, guys, last one. Great job getting through this. This is 3.4 on inheritance. There's some weird formatting going on. I'm not sure what's going on. Oh, there it is. Essentially, the inheritance of gene follows patterns. Okay, again, huge thanks to this guy down here. Awesome slides. They're super helpful, very clear. I like the fact that it's one syllabus statement and then everything is just on that page. Okay, so not as much to review for this one because it's the most recent thing that we've covered, and you have done it in other classes. So the ones you wanted to go over is U1, which is about Mendel, U3, the two alleles, and then A3, which is inheritance of cystic fibrosis and Huntington's disease. There was S2 written up as something you wanted to review, but there's no review needed because that's not something we've done. So you can't review something you haven't done yet. We will be looking at S2, which is looking at predicted and actual outcomes using a chi-square test later. We're going to do it after the test. Okay, it is a good thing to know, very helpful in genetics and in biology in general, but we're not going to do it right now. Okay, so let's talk about Mendel for a minute. So Mendel discovered the principles of inheritance with experiments in which large numbers of pea plants were crossed. So up there on the right-hand side, that's Johann Gregor Mendel. Mendel's principles of inheritance were really key to furthering our understanding of genetic inheritance. So what's really cool is there are a couple of different sources here. So when I post the link to this presentation, this PowerPoint presentation, you're welcome to go in, click on those links, get some more information about it. The biggest thing I want to talk about is kind of why it's important. So he is considered the father of modern genetics. He planted thousands of seeds per trial and carried out lots and lots of trials to make sure that his results worked. This is talking about big sample sizes, yeah? And it's talking about multiple trials. So these kind of things didn't exist. The idea of doing lots and lots of individual tests to make sure it worked, or doing lots and lots of trials to make sure that it always worked, didn't exist until Mendel. And so he kind of not only fathered genetics, but he fathered some of the modern ways of doing science. So that's kind of important. His published work back in 1865 is now considered super, super important. But at the time, everyone ignored his work. He was not thought of as being important in that case. So some quick definitions to review. These are ones that are important, is these ones. So I'm not going to go over each one. You can pause and look at it, but these are the terms. If you're going to talk about inheritance, you've got to understand the terminology, the difference between how to note certain things like carriers and just dominant and recessive is listed here, whereas codominant is going to be like that. All that kind of stuff you need to understand. Okay, you want to talk about U3, which is that the two alleles of each gene separate into different haploid daughter nuclei during meiosis. I feel like if you've gone over the other videos, this should be okay, but just really quick, we know that fertilization involves the fusion of gametes. So the number of chromosomes is doubled at that point. We have 23 in sperm, 23 in egg. The two of those together is 46, 2 copies for each one. So that diploid organism now has 2 alleles for each gene. The alleles are present at a gene locus, can be the same, but they can also be different. Now, they're not going to be exactly the same because of a couple of mutations, but it may not change anything about the protein. Now, meiosis, remember, along with having the chromosomes present in the gametes, it also reduces the number of alleles in each gene locus from 2 down to 1. This is important because this influences how that information is going to be inherited. All right? U6 is looking at the inheritance of cystic fibrosis and Huntington's disease. So for cystic fibrosis, this is a great clinical example that we can talk about. Typically, we're going to use something like pedigree chart to trace family history. So you could do that here and look to see whether or not cystic fibrosis is dominant or recessive. How would you know? Well, you can look here and say, this Punnett square, this must be a recessive characteristic. And the reason for that is you have 2 unaffected parents, and then you have unaffected offspring. So they both have to be carriers. Yeah. In addition, there are two different little videos down here to figure out a little bit more about cystic fibrosis. But the biggest thing to know is that a mutation of the CFTR gene causes secretions like mucus, sweat, and digestive juices, which are usually thin and instead become quite thick. So instead of acting as a lubricant, which is what they do when they're thin, when they get thicker, those secretions will actually block tubes and ducts and passageways, especially in the lungs and the pancreas. So even though we have done a lot with modern medicine to figure out some therapeutic care for lung problems, most CF sufferers will die at a younger age, usually somewhere between 35 and 50. So it is still a very, very difficult disease to live with, and there's not great life expectancy even for those that have great medical care. So let's talk about the actual inheritance. So one, it's recessive, and it's not sex-linked, it's just recessive. How would you look at inheriting this? You could do a cross between a carrier parent and another carrier parent, and you know the offspring ratios with that, right? You would have a 3-to-1 phenotypic ratio. In order to have cystic fibrosis, you would have to have both recessive alleles. So there's only a 25% chance of two carriers having an offspring with CF. Huntington's disease, the kind of cartoony pedigree is on the right-hand side. So Huntington's, or HD, is a brain disorder. It affects people's ability to think, talk, and move. It's caused by a mutation in a gene on the chromosome 4. So a quick review, if you look at this pedigree chart, you can see that this is clearly a dominant trait, right? Individuals are affected when only a single mutated allele is present. This disorder, is it autosomal or sex-linked? It's autosomal because it's on chromosome 4, right? I'm telling you it's on chromosome 4, that's going to tell you it's autosomal. Could you produce a Punnett grid to explain the inheritance pattern? Let's try it. So probability of an unaffected mother and a heterozygous father producing children affected by HD. Now this is going to be a little bit different because remember, unlike, I'm going to go back 2 slides, here in cystic fibrosis, where the mutated allele is the recessive trait, in Huntington's, the dominant characteristic is the mutated allele. So when you cross this, you're going to end up with 50% of the offspring will have the phenotype of Huntington's disease, and 50% will have a normal phenotype or no mutated alleles. So there's a 50% chance of a child with Huntington's disease if you have one parent affected. And this is just a fun little practice. You can figure out, this is called the whirling gene. It's a made-up thing, but it's an activity on thinking about genes as like puzzle pieces in a puzzle. And if this is the pedigree chart, you can actually look at all of the offspring from that pedigree chart to figure out which is the mutated allele or the allele that's causing whirling disease. So you can try that if you really wanted to. That's it. Bionology uses a lot of cool stuff, including iBiology.net, BioNinja, the Oxford Biology textbook. We use the Pearson one, but the Oxford Biology textbook is also very good, and Bob Smullen. So thanks so much for getting through all of those. Good luck on the test, and I'll see you on Monday.